Hi, welcome to the 28th episode of Startup Strides. Today, my guest is Miroslav Gasparek. He's the CEO of Sensible Biotechnologies. Welcome, Miroslav.
Thank you, Javid, and I'm very glad I can be here with you.
No, same. I'm very happy and honored that we're actually making this happen finally. And I'm really excited. I've been looking forward to this episode. I've been following your journey for a while too, so it's really exciting to have you on finally. As I always do before we start, the questions about the company and entrepreneurship and startup, I would like to know more about your background, about your educational and professional experiences. Just, stage is yours. Tell me about yourself in your own words.
Sure. So my background is, I would say, quite not traditional for a life science entrepreneur. So I'm originally from a little village of 400 people called Nosice in northwestern part of Slovakia. When I was a kid, I didn't know anybody in a biotech. I didn't, in fact, even know anybody with a PhD. But I had kind of two passions, and one of them was medicine. The other one was theoretical physics, mathematics, and astrophysics. And throughout my childhood and then my youth, I was like hopelessly torn apart between these two. Am I going to be a theoretical astrophysicist, and am I going to be looking into spectra of the stars far away, or am I going to actually become a doctor? And what I started being fascinated by was kind of application of quantitative methods of physics and mathematics in medicines to better understand and to get a more comprehensive understanding of our human body and its processes, and in the hope that we can heal it. And that's why I ended up going for biomedical engineering. I studied biomedical engineering at Imperial College London, the days I still remember very fondly. And that really opened up the world of possibilities to me, because before that, again, lived in a little village, went to school, which is a kind of a good school, studied in English and so on. So it was bilingual, but there was no strong STEM. And suddenly I had the opportunity to learn from the best academics in the world and the leading experts in a number of areas. So you can imagine that I was bothering the professors quite a bit after lectures and so on. Nevertheless, I became very passionate about the research, ended up doing research in optimal modeling of eczema treatment, systems biology, and control theory during and after year one of my undergrad. And through control theory and the lectures of systems theory, I got to synthetic biology and became really fascinated by applications of computational methods and mathematical modeling and synthetic biology for design of gene circuits. Ended up doing research at Caltech with Richard Murray on that after year two of my undergrad. And because I didn't have enough of it, after year three of my undergrad, I ended up doing research with Drew Endy at Stanford, where in the lab where basically the ginkgo bioworks started. And working with Drew had a very profound impact on me, because Drew taught me to think about my work in a wider dimension. He taught me to think about implications, the societal implications of what I do, and not kind of focus on my little scientific project. I still remember how he explained how I need to start asking question why, and why does it matter? I still try to. And that environment and exuberance of entrepreneurial experience at Stanford really pushed me to become an entrepreneur. And so then I started my PhD just, literally 18 months later with Professor Antonis Papakristodolou at Oxford in one of the best research groups focused on applications of control theory in synthetic biology. And when I was starting my PhD, I already knew I want to be a life science entrepreneur. I told that to my supervisor during the interview. Probably not many supervisors would accept a student with such ambitions, but Antonis did, and I'm very, very grateful to him for kind of all his understanding. I will put it that way. And that was in 2019 when I came to Oxford. When I started, my co-founder, Marian, was finishing his PhD in biochemistry. And when we met, that was kind of the beginning of our journey together, which we've been walking for years now. So, fast forward, Sensible started in 2021, and it's been quite a journey since, but I'm happy to speak about the origins and all of that as we go.
Yeah, I mean, very, very wonderful, interesting resume, honestly. And you mentioned a couple of things, but I'm curious, like looking back, you already said about your experience at Stanford and also your PI at Oxford. But besides that, what have been the most formative experiences that shaped your vision like very clearly throughout your time? Was it Imperial, at Oxford? Was it like classes or mentors or something else?
So there is this little story, like what put real seeds in me was when Professor Jimmy Moore at Imperial College, when I was in the first year of my undergrad, the bioengineering department had this wonderful program or a class called Bioengineering in Action. And what it meant was that lecturers were coming for a lecture, always a different lecturer, was coming in and actually talk to us freshers about their research and its implications for the real life. And that was completely wonderful to me because you suddenly were moving from learning, let's say, a fundamental principles of electrical engineering or cellular systems or triple integrals to suddenly hearing about complex musculoskeletal dynamics, the cochlear implants, the nonlinear systems to describe the cellular behavior, and you could see the practice. And Professor Moore comes in, and in the first sentence he says he's a medical device entrepreneur as well as being an academic. He came and said, "Look, guys, you can do research. It's great. It's wonderful. But as an engineer, your job is not just to make something new. Your job is to make something new, make sure that there are 100 millions of copies of it. It passes the regulatory and actually helps people across the globe. If it only stays on a lab bench, it's not that useful." And that was really, really kind of a formative experience to me. And I just want to kind of give a credit to something that happened much earlier than that in my primary school. When I was around 9 and 10. And the headmaster of that primary school was a teacher of mathematics and physics. And she always really motivated me to go and pursue my passion for maths and physics and so on. And she really allowed me to have very tailored and individual approach to the education. She basically told me, well, you have the great grades. Don't worry about your teachers. You can just go, like, we'll write exams, whatnot. When you're in school, you're getting bored anyways. So take the desk outside of the classroom, pull up your astrophysics work, and just learn that and do the stuff on your own. And not many people would do it. She was already at that time. She was a very experienced teacher. She was just before but really gave me that opportunity, and I learned a couple things. First, like, you need to learn things yourself very often, even because, and I got this as 9 or 10 years old rather than much later. So you lose that fear, you lose that kind of, how to put it, that assistance or kind of walking stick just for thinking that, oh, somebody will teach me this. And second, I learned that it's okay to do things my way, different than the standard way of doing things. And this really helped me a lot, I think. So I don't speak about this as often as I should, but I really wanted to give this credit to Mrs. Pobežalová from a little primary school in Puchov.
I think it's some of those earliest experiences can be the most defining and formative for us, especially in our deep subconscious. So I totally understand where you're coming from. I guess, looking at your background and your, knowledge accumulation and your vision being clarified and defined, you ended up starting your own company. You finally, you always wanted to become an entrepreneur, and you were like, I'm going to start it. And you started Sensible. And how did I want to hear more about Sensible Biotechnologies. How did the idea come about, and what's the core mission and vision behind it?
Sure. So look, the idea behind Sensible Biotechnologies. So it was very interesting way of building a company. So very often, as you would know, what we do as founders of founder-led companies, as technical people, we think of, how do I start a company out of my PhD? Because this is where I've been expert on. This is kind of the hammer that I have, whether it is a wonderful computational toolbox for molecular dynamic simulation, or it is some great gene editing tool or anything else. We have this hammer and we are searching for a nail. Sensible was different. Sensible, where we are building the world's first cell-based platform to design, optimize, and manufacture high-quality RNA to enable the next generation of mRNA medicines, didn't have to do anything with my PhD, which is on control systems theory approaches to synthetic biology, and didn't have anything with my co-founder's PhD, which was on DNA repair in cancer cells. I hope I got that right. And basically, so this is quite interesting because we were transferring the skills, but we really weren't using the same ideas. And we were very problem driven. And in 2021, if you remember, it was still kind of active pandemic world. We still had a bunch of lockdowns all over the globe, but it was the time when mRNA vaccines were being rolled out en masse. And Marianne and I started thinking, look, mRNA, well, clearly it works, and it's probably going to be a huge business. It's going to be a huge market. And we started thinking about how the mRNA is made. One of our friends at the time, she basically said, "Well, I'm working on this mRNA startup, and I make RNA using this thing called in vitro transcription. It's cell-free reaction, just make RNA this way, and it sucks. It's cumbersome. It is expensive. It's not great." And she was like, "Oh, what if you try to do it in cells? Like you guys been thinking about working with cells. What if you try to do this in cells?" And we had a lot of other ideas. We were really kind of problem driven, but we were narrowing probably from 20 ideas. We were taking this a lot to experienced people, and we set up a number of categories from like what is our team idea fit to market size, and we were narrowing it down, and this mRNA in cells was resonating, and there were a couple interesting things. So first of all, what was a great indication was that people thought that mRNA is a big market, or that actually makes sense. And at that time, we were really thinking more about just making RNA differently, making it cheaper originally. But as we were building up and talking to more experts and leaders in the field, we started understanding that the problems of RNA go far kind of beyond just making mRNA cheaper with simple logistics. But we need better, less immunogenic, more functional, optimally designed RNA with great dynamics and so on. It was clear that this resonates with the people. We also saw that there's we somehow thought that somebody must have done it and must have failed. Moderna will have run 20 internal programs on it, or BioNTech will have run 20 internal programs on it. And actually, it started to become clear that not really. People didn't think about it. And that was one of the most frequent questions was, has anybody done it? But why hasn't anybody done it? And we were like, well, maybe people didn't really care that much about mRNA before we got assets approved. It were just a couple of companies really looking into this deeply. Obviously, there were deals in the space between the big companies, even before 2020. But that's when it really took off. And all in all, we saw a couple things. We saw that, okay, looks like a meaningful problem, looks like a problem we can easily communicate, looks like nobody has really done this, and looks like it's orthogonal and interesting enough. And this was, I think, something that we really thought is interesting, that these people said, this is an interesting problem to work on. And it's very, I think, important, not necessarily just for you as an entrepreneur to work on something that matters, but what you want to make sure is that the people are excited by your idea. And hopefully, if they are excited by your idea, they are more prone to work with you and actually engage actively. If it's not 28th iteration on this or that. So I think this is a great thing. And probably now I should explain more in depth what we do at Sensible and just how this differ from the different approaches.
Yeah, I actually have two questions here related to what you said. And one is, well, obviously you touched upon COVID, and as I feel like I really started learning about mRNA and its application during the COVID vaccine time. I mean, my undergrad is in science, but I wouldn't consider myself like a scientist. So if somebody has been in academia for a while, they probably heard about it for some time before that. But I feel like, as you said, it really took off during the COVID time and became this mainstream thing. So my first question is, so before COVID, mRNA technology wasn't widely discussed in the context of vaccines or therapeutics. In your opinion, why has the therapeutic potential for RNA gained so much attention only relatively recently? And why haven't we really seen any mRNA medicines approved outside of infectious disease vaccines?
So let me actually just kind of reassert this statement. So I think mRNA, in its essence, I would say probably it was first really suggested as a therapeutic modality. You would need to get somebody who has kind of the full picture, like Andy Gill, on the podcast to kind of tell you the all origin stories. But the first mRNA studies in vivo, I think, were on rats in 1992. I'm not sure, but I think it was enzyme replacement therapy, right? So the first thinking was around that. And even before COVID, if you look at a number of assets that these leading companies like CureVac, Moderna, and BioNTech had in the space, very often it were cancer, or people say cancer vaccines, you should really say cancer immunotherapies. And these were the assets like enzyme replacement therapies, because what is the most natural thing? We think of the mRNA. mRNA gives me instructions for a missing protein. What am I going to do with that? I'm going to take mRNA, hopefully get it to the body, and then basically, hopefully that mRNA provides the instructions for the missing protein. But exactly as you've said, we have not seen any mRNA therapeutic modality other than the COVID vaccines and really some other infectious disease vaccines approved. And I think there is a couple of reasons, and different people might have different opinions. I think that we obviously saw the challenges with the delivery, like that's got significantly improved over the last couple of years. And I mean, more than the last couple of years, it was the real kind of invention and improvements in the lipid nanoparticles. So that was definitely one thing. The other important thing seems to be the, say for the cancer immunotherapies, the better neoantigen selection and kind of discovery, right? Picking up the right neoantigens to direct the mRNA for. The other thing, though, is how do we make mRNA that has really great quality? And there was so many advances in the last couple of years, even you could say decades, right? If you think of cappings, so the modification at 5' end of the mRNA that's required for appropriate translation and so on. So invention of Cap1 enzyme, it was modified bases, right? So instead of using uridines, using pseudouridines or environmental pseudouridines or 5-O-methoxyuridines to reduce the immune response to the mRNA, and also the understanding of the mRNA design. But what we think is that in fundament, the way, for example, we make RNA hasn't really changed that much. The principles are the same. mRNA is made in a way of making in vitro transcribed RNA and then packaging into lipid nanoparticles and supplying that. And we think that this method has inherent limitations, say on making very pure RNA, making that scalably, achieving that purity, especially from double-stranded RNA at a large scale. At the same time, making sure that mRNA is modified more naturally, similar in a way that mRNA in our cell is, which is not like 100% pseudouridines. And it's not 100% uridines. There are like site-specific modifications, and many of them. And ensuring that we can make mRNA, as one of the mRNA luminaries in the field told me, as close as possible to the native human mRNA to make it more likely to work well for the therapeutics. Will this work? We will see. That's what we are working on, making completely orthogonal way of making mRNA that has higher quality, has, say, reduced immunogenicity, improved translation efficacy, where we can design its almost expression dynamics well. That's kind of at the output, and at the input, we want to make sure that this mRNA doesn't have a super complex supply chain and can be made in a way that is fast and scalable and in a way that is cost efficient.
You actually kind of answered the follow-up question, which was going to be what does Sensible do differently compared to traditional production methods? But you said, you guys trying to, I mean, when you go on your website, one of the first things you see is like a scalable production of RNA in a more pure, less immunogenic form. So that was my main question, is like, how does it differ from traditional? But I think you kind of like answered it, but feel free to add to that if you.
Yeah, so I'll actually add one thing. And what I would add is, I think that it's kind of fundamentally orthogonal. If we make mRNA using in vitro transcription, what we need to do is to take the DNA template, have a circular plasmid DNA, transform it to the bacteria, grow bacteria, purify out the DNA, linearize it, and then run this in vitro transcription. So you take this DNA, you add a number of enzymes, capping enzymes, T7 RNA polymerases like T7 RNA polymerase, that nucleotides, cofactors, and so on and so forth. and then this makes the mRNA, and then you purify the mRNA out, and you purify it again. Methods like chromatography, oligo-DT, capture, and so on and so forth. And right, there's a couple of things. So you have a good control over that reaction, for example, right? Because you know what's going in. That's a great advantage. But also, well, the constituents you use are quite different, say, than what, again, our cells would do. So if you think of it, like cells are using complex multi-subunit eukaryotic polymerases to make mRNA. D7 polymerase predominantly being used now. Well, it's actually an enzyme we can make it in a clinical grade quality. So obviously, we use that one, but it has a number of challenges, and people are innovating on that. They're trying to directly evolve it to reduce the double-stranded RNA content or improve kind of the length and transcribe and so on. But it's something which we think, why don't we do a radical change? Why, if you make mRNA in cells, you don't need to worry about capping because cell will cap it. You don't need to worry about how am I going to get a poly A tail on? Well, cell will add poly A tail to its transcript. Obviously, you have different challenges, and I don't want to get into the weeds of our technology and how do you overcome some of these challenges. But we're glad we are overcoming them, and we think it's a very exciting orthogonal platform. And what makes me really happy is that even when we talk to the top people in the mRNA field, they get excited, they get curious, and I think for an early stage company like us, because we are still an early stage company, this is among the most important thing for people to get really excited about what we do.
If I'm wrong, you've raised over $10 million, and you also formed strategic partnerships with large companies like Ginkgo Bioworks, and doing that out while being based in Europe, right? Like usually you see this larger kind of like VC interest when you're usually based in like Boston or San Francisco, but you're doing it out of Europe, which is also an accomplishment itself. It looks like you guys are answering a need in the market, and that's why so many VCs are interested. How does this, like strategic partnerships come out? This, the VC interest come about, and how's they shaping the development of sensible biotechnologies?
So that's a great question. I think what helped us a lot was going through Y Combinator. That was also a really transformational experience. It really forces you to get the basics right, which I think is so important. Like talk to your customers, start a company as a Delaware C Corp, which means it's much easier for the US investors to put the money in. Would you want to invest in a little obscure Slovak entity, společnost s ručeným obmedzením, limited liability? You'll be like, the hell, I'm not giving whatever $20 million to who knows if it even exists. Right. So, but if you're a Delaware C Corp, and this is what a lot of my friends from Central and Eastern Europe and Europe have done, start a U.S. company where people have kind of a clear understanding what's going on. Then, and you can have a subsidiary 1 to 50, but you get a good corporate structure. And I think that was one of the things that helped us. The other thing is, so I think as a good CEO, and I started doing this rather early, I just try to build network and relationships in the US, East Coast, West Coast. And I don't like to say networking. I don't think this is I don't call this networking because networking is kind of, we imagine you get your kind of name tag saying hi, I'm Jimmy, and you go and say everybody, hi, I'm Jimmy, and why? No, it's more about so okay, these people are relevant to what I'm doing, or they're really smart, just really smart guys, really smart people, or and men and women, and you go and ask them hey, it was a great conversation, who else should I talk with? Or they come up and say, oh, I should introduce you to this person and that person. And that's how you start forming these webs, and how you start forming ideally a closer relationship. So and I'm very, very blessed to have some wonderful mentors who, some of them I've met originally serendipitously, but you then start really meeting people through introductions, and that's I think which starts becoming really, really helpful. And the reason why I'm speaking about it is that I think this was one of the reasons that we're able to partner with Ginkgo Bioworks early on. I mean, obviously we can't speak about the other partnerships, but once this kind of puts you on a map, we're a very young company. When we did this and announced it, it really helped us to get some good media coverage in Wall Street Journal and Endpoints and for US Forbes, and it basically allowed us to really, really start building up that credibility. And then somebody well connected suddenly writes you on Twitter, just a very simple thing. And you reply, you meet in Boston, you start building that connection. Later on, the person can angel invest, and then they start becoming really helpful. And I think it was about building up the reach, also building up the trust, and making sure that ideally when you say you do X, you do it. When you say Y, you do it. Ideally having zero ego, because look, I'm a first-time entrepreneur. I have so much to learn. That's, I think, very important to keep, that you're there, eager to learn, eager to listen. So yeah, that's I'd say are some
Got
of
it.
the core things.
Got it. And you also touched upon this here, as you did. The potential for mRNA extends well beyond vaccines. I mean, 2020 saw its application mostly in the vaccine area, but you already said something about cancer immunotherapies and etc. How do you see its application evolving, and what broader impact do you foresee on biotech and pharmaceutical industries?
So I think what we are kind of seeing. So I like to think there was a generation one mRNA medicines. These were those earliest trials with unmodified, say, mRNA and so on. And then there was generation two, which kind of gave us COVID vaccines, modified Cap1 mRNA, really working well, for that purpose. But I think now we are looking into very exciting applications as a field. Genetic medicines, right? For example, in vivo gene editing, encoding gene editors and base editors and prime editors through mRNA, doing ex vivo cell therapy, re-engineering cells using mRNA, again, encoding transcription factors, so on. Another exciting thing to me is the applications, obviously, in cancer, and not just necessarily cancer. As Professor Jeff Kohler from Johns Hopkins says, don't call it cancer vaccines, call it cancer neoantigen therapy. Kind of not just looking into vaccine applications, but also, say, encoding actual therapeutic molecules into mRNA. And I think for all of this, we need different mRNA properties. We need to be able to dose mRNA high in a number of these cases, up to 1,000 times per dose of the typical infectious disease vaccine. You might need to dose repeatedly, especially for enzyme replacement therapies. You might want to shape the dynamics differently. And for that, you need very highly functional non-immunogenic RNA, and obviously would like it to be available as fast as possible. And a big vision for Sensible is, can we have this modular production of high quality RNA? And I will say design, optimization, and production distributed and enabled locally. So we don't have just, couple, 1, 2, 3 big hubs in Europe, and suddenly a pandemic comes and then we close the borders and suddenly we start all this, I shouldn't call it vaccine wars, but we start all of this, hassles and discussions about appropriation versus what if every region could have its own modular mRNA production facility, which in the biological peacetime produces mRNA cancer treatments, and in biological, I call it like wartime, so war on pandemics and infections, produces large amounts of vaccines against these ailments. And I think that this is a very exciting element of Sensible. How can we basically build out that distributed mRNA manufacturing capabilities while designing and making the best-in-class mRNA and partnering with our pharma partners on accomplishing that?
This actually ties perfectly to my other question, which I know, and I've read a few things you've written before. You're quite involved and advocate for biosecurity and biosafety efforts. And again, you perfectly touched upon that. During the pandemic, there was a lot of politicization of vaccines and who had access to what, and that created a huge power imbalance. So my question is, what role do you see biosecurity playing in the future of biotech, and particularly how does accessibility to mRNA production fit into this picture?
Right. So I think people didn't think too much about, let me retract that. A lot of extremely smart people have been thinking about biosecurity deeply for years. And biosecurity thinking has been evolving because 50, 60, 70 years ago, the biosecurity rate was about how do we make sure that somebody doesn't sprinkle
Anthrax.
weaponized anthrax and
Yeah.
I don't know, variola and things like that. And it could go even further, like 500 or 600 years ago. If you read like an excellent book by Neil Ferguson called Doom, you will learn that in 14th, 15th century, right? That's when they invented a quarantine. The ship was coming to the port in Italy, and they said, okay, you're not getting off that ship for 40 days. And if you guys don't all die, then you can come in. Not sure it was completely effective. Presumably helped a bit, because not necessarily sure they got rid of all the rats, for example, and pests that could spread ailment. But this is just kind of a side note to this. I think that it has been evolving, and today we are looking at a number of different problems in biosecurity space, not just looking at evil state actors, but looking at challenges of distributed bioengineering, and democratization of bioengineering can go in a good way, and it can go in bad way and in the ugly way. So I think that's a number of challenges. So how do you ensure the surveillance? How do you ensure the rapid response and recognition? And how do you ensure kind of a long-term response? So these are, I think, among the core principles, right? If there is, say, a pandemic, or God save us, an engineered pandemic, right, in the future. First of all, how are we going to learn about it fast? Because once it kind of all spreads, that's a challenge. The second thing is, okay, what is going to be the rapid response? At first, how do you utilize the non-pharmacological interventions? And, I worked with the Ministry of Health in Slovakia early on during the pandemic. I then worked with Palmer at Stanford on some biosecurity questions, and ended up advising a board member of WHO on some biosafety and biosecurity questions. And we were kind of thinking about planning these things as we went, and especially planning for a small country. And then you have a third thing, which is like, okay, so how do you ensure that you have a sufficient capacity and capability to make pharmacological countermeasures like mRNA vaccines? But it can be other things. Like, it's not just about vaccines. Like, if you think in the pandemic, suddenly we didn't have enough masks, and we didn't have enough personal protection equipment, because nobody was ready for that. And suddenly we learned that our supply chains are broken. We were shipping it all from Asia, which was totally fine up until a certain point. And suddenly you learned, okay, but damn, we don't make these things here, and the planes are not flying anymore. And I think that this is an interesting problem. Say in the European Union, we have Schengen area. Well, in Schengen area, it means like there is no border controls, Slovakia to Austria, Austria to Germany, or anything like that, really. So you can, once you get a lorry, and I'm like exaggerating, the lorry full of like whatever weaponized pathogen to Czech Republic, you can kind of drive it around. Maybe if it's a lorry, they will like check it at some point. But if it's just a car, and okay, so this, and I'm glad that we don't have these borders, because we can then travel and so on, and it's very easy and convenient. But it also brings in the risks, and if suddenly during the pandemic, we went from a complete freedom of movement to absurd restriction. I mean, sorry, let me retract that. I don't want to say absurd, but absolute restriction, right? We went from, hey, I want to go to the UK, so I'm literally going to the airport, buy a flight ticket, I show them my passport, and I go, to you can't leave your house. So literally being confined in 20 meters squared. And it was probably necessary at that point, obviously, but we need to balance that. Kind of open borders can help us to have a great time, can help us to spread the pandemics. And at the same time, we don't want to go back to being closed at 20 meters square. The resolution for that is like enable the countries to have distributed, decentralized capacity to make medical countermeasures like mRNA vaccines, like even other things like needles and personal protection equipment, so that if there are any such challenges, we can basically be ready and start, kicking off the production not in a couple of years, but in a couple of hundreds of days, or even a couple of, obviously after we finish the proper clinical testing. So that's yet another thing. But that wasn't, I would say, the biggest bottleneck of this pandemic, right? During the COVID, mRNA vaccines were designed in days. It was tested in months in clinical trials, but it took almost until infinity. We weren't able to vaccinate everybody who would have wanted, and especially in the economically developing countries, before the pandemic actually ended. And I think we need to change that.
That's very fair. We are coming towards our end of our episode. I have a couple more questions. One, want to follow up on what you just said, and I think there's always, not always, but you often see a bit of a discrepancy between the public and private sector. And there's some kind of like a miscommunication or perhaps miscoordination. What role do you think both the public and private sector should play in developing and implementing meaningful biosecurity solutions?
I think this is such an excellent and critical question. We can't, so biosecurity and biosafety, right? That's a great extent about regulations and enforcing regulations. This is not something that Indian private sector can put its internal regulation, but you can't. If you're Moderna, you can't come and tell somebody like, no, you need to use this kind of disinfection protocol, right? The governments can do it, and international bodies. So we need the governments and private sector to work together on the biosafety and biosecurity regulations. That's kind of the thing number one. The governments are also there to, I think, they play a very important role on providing the demand for these solutions. Because look, we haven't made that many vaccines in the last couple of decades because it doesn't, it's not profitable venture really. And I think like what we need the governments to realize is like, okay, we actually need to invest in say securing the demand so that people can build up the supply, because sometimes the technical aspects are not the bottleneck. It's the economical aspects that are the bottleneck. So that's kind of one thing. I would say the other thing is, and this is especially important for smaller companies, but also for bigger companies, I think, it's the help with building infrastructures. VCs will happily finance us building cutting-edge, high-throughput screening platform to design better RNA and better RNA sequences. They will happily finance us to do clinical development. But if I would come and say, hello, I'd like to build a factory. I'd like to spend $25 million like CapEx and the development, they won't be too happy, or many of them won't be too happy. So the reason why I'm saying that is like, this is where the public sector should step in, as in many countries it does, and basically come and say, and okay, look, this is a very promising technology. It's very interesting. Why don't we support you to build this out? And it's actually something that, at Sensible, I would say we are having a very reasonably good experience, talking to multiple governments, which I can't disclose which ones, but basically the people are willing to listen. And you need to obviously explain it in the relevant context. Talk about job creation, you talk about economic impact, you talk about health impact, you need to talk about security impact. But I think like the governments have played a crucial role in providing the proper biosecurity framework, and international bodies as well. Because here is the thing, like viruses don't care about the borders, right? Sure. If you have a war, it's like, this is my ally, this is not my ally, so I'm going to attack this other dude. Viruses are not that smart. They don't care about diplomacy, geopolitical treaties. They're like, I want to replicate. That's it. That's the purpose. I want to replicate. Where? As long as I can, I don't care. So that's, I think, something we need to bear in mind and work together better.
This was a really interesting episode. You're full of insights and wisdom. I really appreciate this episode. I guess we are at the end. I don't want to take too much of your time. One question I have, as we wrap up, actually, there are two questions combined in one. One, what's the long-term vision you have for Sensible, like in the coming 5 to 10 years? And the second one, what advice would you give to scientists, researchers, especially based out of Europe, who are trying to move their research from lab to market and become entrepreneurs?
Let me answer the second question first, because I want to end up on the note of Sensible's long-term vision. My advice would be, first of all, like if people are telling you it's X or Y is impossible, like, don't listen to them. Take feedback and ask why, but don't listen to naysayers. What I would think the practical advice I would give people is like learn, and don't learn just about the science, like try to understand the bigger context. You need to start developing fundamental understanding of capital markets, publics and private, the mechanisms of investing, understand what's going on in the industry, right? Are the engineered bacterial therapeutics what flies now, or is it antibody drug conjugates, or is it siRNA? Like understand what's going on in the world. And I would say build up and find great mentors. I've been very grateful for some wonderful mentors and people I can learn from, whether it is somebody like Florian Schuster, who co-founded BitBio, Jason Foster, who leads Ori Biotech, Jason Kelly at Ginkgo Bioworks, and a number of other mentors I can't even name, that helped me to shape my thinking. Also, I would say, our lead investor for our seed round, Vishal Gulati from Recode Ventures, really have a great relationship. What I'm saying, this is like, don't have high ego, so have very low ego, but also think for yourself. It doesn't mean that if somebody's telling you something, it must always be true, but think of why people tell you a certain advice, and this will help transform you. And I still have a long way to go on that journey, but hopefully into a mature, thoughtful life science executive. What is the Sensible's vision? I will say this. Look, my vision for Sensible really is to, in 10 years, I'd like to see the cell-based, kind of designed, optimized, and cell-made mRNA medicines made and partnered with Sensible to be reaching the market and making the change in the world. I'd like Sensible to be fully integrated mRNA company that will help our partners and pharma companies to bring the medicines across the globe. And if in 10 years I will look across the globe, I'd like to see multiple modular biocontainers with Sensible's logos churning out mRNA that will save people's lives. So that's my vision.
Thank you so much again for your time. I wish all the best to you with everything. I'll continue to follow up your success stories on LinkedIn and everywhere else. And I hope we can stay in touch too. But really grateful for your time. And thank you again.
Yeah, I'd love that. And Javid, thank you so much for all the great work you're doing on a podcast. So folks, if you haven't heard the other episodes, go, because Javid is making such a great job of getting great people and great content on the life science side to all of us.
Thank you so much, Miro.